So the today's topic is like intertwined metabolism and epigenetics unveil the way for therapeutic intervention in gastrointestinal cancer. I don't know why I went into field of gastrointestinal cancer. That was the interest from day one. I joined Cancer Research Institute in 99. Started with liver cancer, gone much basic understanding into a gastric cancer. And these two cancers have given me a lot to understand. We started working in the beginning in 2000 on human cancers directly in the field of liver. And then slowly from finding some of the very interesting findings there, we want to decipher the mechanism first. Why this is happening there in a human tissues. And then while understanding, we came out why it is so important and that can have a high therapeutic relevance. So you have to say change. Okay, change. So today's topic, if you say I've written topic for today's interaction, it is not a one-way flow. You can stop me anytime in between, and that will help us to share what you are not understanding and what I'm trying to communicate with you. Fundamentals of epigenetics, how epigenetics is so closely linked with gene expression. And the third topic is very important, epigenetics, metabolism, and lifestyle. We all talk about epigenetics, but we don't know how it interacts directly with an environment today. And that interaction with an environment brings about a lot of changes in our body, and which brings a lot of gene expression alterations, and thereby changes our whole phenotypic factors. And then in the end, I will be talking somewhat about our own science, what we have done at Tata Memorial Center, and how it has gone with a clinical relevance. So these are the four topics, but the main topic is the today's interaction. You have to remember, we have to interact more so that when I'm giving a talk, talking to you all, I can send back some knowledge to you, which you can try to grill into it and see whether you can take the field much ahead into it. Please change the thing. So this was the Times cover, magazine cover, which takes why your DNA is not your destiny. And the below it writes, the new science of epigenetics reveals how choices we make can change our genes and those of our kids. So what you are doing today, it is not you which you will change yourself. You will change the nature and other disease associated with the next generation which will be coming into picture. So all the epigenetic changes are heritable. They move from one generation to another. So if you are harming yourself, your epigenetics will be totally altered and that will be transmitted to the next generation. And it is not generally we believe females are responsible for transmission of maximum factors. But now the studies are available where people have shown smoking, tobacco chewing, and other type of environmental factors completely change the sperm epigenome. And if the sperm epigenome is altered, it results into a lot of infertility issues, a lot of diseases associated when the kids are born. So it is not always the female, it is more the male, because the chromatin which is condensed in spermatogenesis is very, very essential for a proper fertilization. If during spermatogenesis, if your epigenome is not condensed, then your loss of mobility happens. So it is important in both the perspective to look into how epigenome affects the embryonic development also. And why this becomes very important when you look from genome to epigenome changing your own genetic destiny. See, if you see from here, what we all talk about, there are more than 25,000 genes, which makes mRNA and further protein, but the genome remains the same at a stage of zygote also. This is a single genome. If you see, when you have a multicellular organism like humans, we have one genome, but we have more than 200 cell types. Within a liver, we have 7 different cell types. So you imagine you have more than cell types, but still the genome remains the same. How? Then liver functions, how stomach functions. The functions are totally different. And there comes the role of, we have believed today, there are 200 epigenomes. And every cell, like see, the genes which are expressed in liver may not be expressed in brain. They may not be expressed in kidney. And the best known example, if you see, the gene which is the, you all know about liver cancer maybe a little bit, the known marker for liver cancer is alpha fetoprotein. That's the best known serum biomarker today in the field of cancer. But this is not expressed in the adult tissues like us. But in embryonic tissue, this mRNA is, the mRNA and the protein alpha feto is made. But anyhow, if this protein is made in an adult, it is a result toward developing a cancer of liver. So therefore, these things becomes very, very important to understand. And how this differential gene expression is regulated is a DNA methylation phenomenon. Yes. And so what I was talking to you, epigenetics take tags from parents to offspring. I will talk about the tags a little bit more. Epigenome alters by environment and nutritional condition, which I just talked to you. Epigenetic mechanism prevent delay or favor the onset of health problems and quality of life. So these are the major properties which I will be discussing a little bit later about how they play a role in there. No, next, earlier one. Yeah. So therefore, what the player important is gene environment interactions influence epigenome. And not what I was talking about, multiple phenotypes are originating from a single genome. You talk about the cancer, you talk about the blood, the sperm, and other cellular types, all are originating from a single genome. So the difference only comes via epigenome. So just to you, for your basic understanding, what is epigenetics? Epi always means above or over. So what it says, genetics is the same word which you all have studied in depth, but anything which is above the genetics, we refer as them as epigenetics. Who discovered it a long back, 1942? Conrad Waddington coined the term epigenetics, coming from epigenesis. Heritable changes in expression without change to underlying DNA sequence, a change in phenotype without a change in genotype. So what you are doing is, there is no change in the genotype or there is no genomic mutations, but are different. I will give you a very nice examples about how you can really see and differ these phenomena. Next, please. Next. Natural occurrence, but influenced by factors like age, extrinsic and intrinsic environment. What I'm talking about extrinsic was the environmental factors. The major environment intrinsic also plays a very important role, is our own mental status, our own hormone levels. These factors also dictate our own epigenome and therefore our mental status. People say we are in depression. Why we are in depression? Some hormones levels have altered. And why hormones levels? Therefore we feel depressed and our expression changes. So these basically comes under the factors of intrinsic environments. Our own lifestyle. Our own lifestyle. Why I say lifestyle are important? Generally outside the colleges, if you see, I will give you a very nice example. There are roasted paneer, roasted chickens are available outside the colleges on different thelas and shops. But when you go to a hotel, 3-star or above, these roasted chicken or a roasted paneer are served with a lot of green leafy vegetables like onion, carrot, lemon. So why they serve, what's the difference between the two foods? Any thought process on that? Yeah? No, they are never served for a presentation. No hotel will try to put a food for presentation. See, when you eat this roasted food, this blackening or a browning is rich in nitrosamines, nitrates. And when you eat this nitrosamine, they can convert into your own liver into a free radicals like nitrites and nitrates. So when you are eating the green leafy vegetables like cabbage, carrot, onion, or a lemon, they are rich in antioxidants. So whatever the free radicals you are generating in your own body are taken care by these antioxidants and they are absorbed. So this gives a very good balance of generation of the free radical and the absorbance of the free radicals by the antioxidant. So your body is not under harm, anything's happening. And these free radicals are the major cause of damages within our own body at DNA, RNA, and protein levels. So there was a thing which was talking about tag. This you all must be knowing very well. We all have a textbook figure from genome to epigenome. I'll try to convince you. Central dogma. DNA can make DNA. DNA can make RNA. RNA can make protein. So this we all know, and this is a central dogma you must have studied in your textbook. But today the central dogma has been changed to epigenome. See, this DNA cannot make RNA if there is a histone methylation, if there is a DNA methylation happening. You know what is DNA methylation? Someone has studied about? The cytosine of the DNA undergoes a methylation at site 5. And if it gets methylated, those genes will never make mRNA. And if they don't make RNA, those RNA, so this comes as a differential thing. So whole DNA cannot make RNA. Only 5% of a total DNA makes RNA, which is transcriptionally active. 95% of a DNA is thought to be a junk DNA, what we know today, which lies in a facultative heterochromatin state. Similarly, RNA has also gone under methylation at a site adenine. Who all are maybe the microbiology student and studying bacteria? In bacteria, the methylation happens on which nucleotide? Cytosine or adenine? In bacteria. Little louder, what's the problem? No, in bacteria it happens on adenine. That's the difference between the eukaryote and a prokaryotes. So if this methylation happens, then these RNA becomes more stable, and if they become more stable, they will make more protein. So under a lot of stress phenomena, people are trying to find out in mangroves where the salt concentrations are high, these methylation levels are very high. So this stabilizes the mRNA, and these mRNA last longer in the system and makes more protein. So that studies are emerging in how stress transcriptionally, the DNA cannot make RNA, but still the protein levels are present. And why is it present? Because the mRNA is getting methylated under stress and thereby continuously go on making the protein. And then there comes the third tag is the methylation or the phosphorylation or acetylation of one of the protein, which is called histone, but is not the only histone. All the protein, whatever the pathways you must have studied till now, undergoes a very natural modification, which is phosphorylation, to make them active or inactive. So basic, now the central dogma, people are talking more in detail about how these things regulate gene expression and your own phenotype. So now I've just summarized you here. So this is a very textbook figure from standard 6th or 7th now. There is a nucleosome, which is made up of 8 histone proteins. Then you have a euchromatic 10 nanometer fiber. It makes 30 nanometer fiber and then a metaphasic chromosome, a very standard figure. But the most important thing which happens is the post-translational modification on these histone proteins, which converts this highly condensed chromatin into an open organization. I will talk a little bit more about it. Then comes, I've talked to you about the tags of RNA and DNA just now. I talked about histone. And there's a third phenomena, which is also very important, is methylation, is the microRNA, which I will not be covering today in much detail about these. I will be going in much deeper about understanding of, because this is a huge topic of epigenetics. If I try to cover and give you an overview of all, it will be too much for you all. So this will be the topic of debate today. See, this is my DNA. When it has been sequenced, when we sequenced our human genome, when the sequence came of whole human genome, sequence, and a student, I should keep some prizes here next time. 2002, we get our complete human genome sequence. And we thought we have gained everything. So this is present in a DNA. But when you read any book, you tend to put a lot of tags into your book. And every book has an index. Why you put tags in your book or why you highlight with a highlighter? What's the purpose of it? That part is important. And why you want to put a tag? Because that part is important and you want to open it anytime you want to open it directly on the same page. If I'm going for an exam, I will put up all the red tags. These tags are to be opened whenever I want them. So what the same thing what the DNA does, it puts multiple tags in the genome and it knows if the environmental or the intrinsic factors are like this, I have to open this gene for expression immediately. So these are all the red tags which need to be opened. There will be few other class tags which need to be repressed. So it will immediately stop the transcription and stop the synthesis of those genes. So these other tags becomes very, very important in our whole human genome also. So just a basic fundamental, because when we talk of any kind of a biology, always chemistry comes into picture. And therefore, we generally say biology and chemistry together as a biochemistry. That's very important to understand. So we have a double-stranded DNA. The backbone is a phosphate. And why this DNA and histone interact? Why can't DNA interact with any other protein within a nucleus? We have thousands of protein within a nucleus and cytoplasm, but DNA always interacts with histone protein. So what is your thought process? What you all feel, why that interaction should happen? Why DNA interacts with histone proteins? Who is there? Little louder. Very good. So that's the chemistry here involved. If you have a positive and a negative charge, they attract each other. So, but if they attract each other, that will form a highly condensed organization. And that what happens, it forms a highly condensed organization. Because the two amino acids which histones are rich in are lysine and arginine. They are basically the basic amino acids. DNA has a phosphate backbone. So they come together and attract each other and combines a complete nucleosomal organizations. And so if these histones are coming, then they cannot make RNA. I told you, if they are 30 nanometer fibers or a metaphasic chromosome, they can't make RNA. So different groups are attached to this lysine and other amino acids. Phosphate are attached to which amino acid? Phosphorylation happens on which amino acids? If you are not asking, I am asking you something. Which amino acid undergoes phosphorylation? Serine, tyrosine, and threonine. These are the three amino acids where the phosphorylation takes place. And ubiquitin happens on lysine, acetylation on lysine, methylation also happens on lysine and arginine. What they do, they are reversible and highly dynamic. I will talk about how they are added. So basically, all amino histones are positively charged. So they are neutralized by these addition of amino acids. If I am neutralizing a positive charge, the negative charge is there, so they start repelling them each. When they repel it, what happens, so chromatin is like this, it opens like this. If each finger is a nucleosome. And favors the recruitment of cofactors. And what happens finally, if you have a highly condensed organization, the DNA becomes loosely packed when these N-terminal tails of histones get some charge. So how these histone modifications happen? So there are three things, a pencil and a rubber and a notebook to read. The pencils are generally we all say as the writers. So enzymes are added on the histone protein by few writers. Erasers rub them out, remove them. And there somebody has to read these marks. When the marks are added, these are read by the readers and there are specific domains which are of not much interest to you. This does not talk about if the lysine undergoes modifications, it is done by histone acetyl transferase. Acetyl group is transferred to the lysine, it is called acetylation and done by the hat. And there are different modifications which are happening, which will not much interest to you. So if I summarize at this point of time the basic fundamentals which I talked to you, this is the lysine. If I add an histone acetyl transferase and there is a acetyl group, it is transferred and forms N-acetyl lysine on any protein. So what I am doing, I am neutralizing the positive amino charge by the acetyl group. When I'm neutralizing it, what I am doing basically, the highly condensed chromatin organization, if all the charges, if you see, are come up here, there the DNA loops out and it completely opens. And this opened chromatin is a site where a lot of proteins like Tata binding proteins and others bind and initiates the gene transcriptions. So this is the, if you see, this is the whole part is a hardcore chemistry to understand, and this is what the biologists generally define this phenomena. So this much what I have just now talked to you. So overall, if you see, the condensed chromatin cannot make RNA, inactive methylated cytosine, deacetylated histone. Active open chromatin, unmethylated cytosine, acetylated histone, they are open, they can make mRNA. So these changes, these dynamic changes convert and open into a condensed and thereby dictate whether the transcription has to happen or not to happen. See, this is cell cycle. Why I'm showing you a cell cycle? What are the different phases of cell cycle? Somebody from this corner? What are the different phases? Written on the board, you can just repeat it. Huh? Yeah. So what's the difference there in all these cell cycle phases? What is happening? Cell is dividing from 1 to 2 cells. That is one of the basic function of a cell cycle. But if you see the difference, the G1 is a functional cell, it's a highly open chromatin organization. The same cell, when undergoes into a mitotic phase, it have a condensed chromatin organization. So if you just go back. अच्छा इससे हो रहा है क्या? Oh, thank you. So if you see in an M phase, it has a condensed metaphasic chromosome. In a G1, it is a highly open chromatin organization. It is replicating into 2. And again in G2, it is getting condensed to form a metaphasic chromosome. So same DNA, depending on the need and the environmental factors, try to divide or not to divide. So these are two identical twins. What is this? Monozygotic twins. Okay, coming from what are anybody can define for me what is monozygotic twins? One can be little louder here. Single egg or a zygote? Yes, from a single zygote. But when you see, so they have 100% same genome. So you agree with me. But whether they are 100% identical, after a few years, 2 years, 3 years, whether the two identical or a monozygotic twins, in terms of their behavior, in terms of their nature, in terms of their dietary habits, they're 100% identical. Yes or no? No. So why so again? If they're coming from the same zygote, they should have 100% identical behavior. But still they don't have. And why they don't have? The research has clearly shown with the influence of a different relationships among themselves, with their parents, with the environment, their DNA methylation is completely becomes different. And if you start, people have shown nicely the blood sample studies from month to 3 years. In 3 years, their epigenome becomes totally different, and therefore their behavior starts changing. And that has been shown. So identical DNA sequence, but epigenomes are enormously from diet and the social environment. So therefore, when you talk of a science along with environment, another factor I'm putting up here, which is called nurturing. So how the kids are nurtured also we change their own epigenome, and thereby their behavior and other factors changes. What is this figure? A bee's royal diet. Identical DNA sequence but epigenomes. So if you see, all queen bee are the workers coming from a same genome, from a same mother. But still, their physiology, Their anatomical structure of uterus, everything is different. Why so? What is happening? Anybody has studied or given a thought on it? So if you see, queen's bee larva are raised in a cup filled with royal jelly. So just this is the difference. The queen bee diet is rich in royal jelly. Royal jelly compound triggers epigenetic changes like DNA methylation and stone modification, altering gene expression leading to a distinct phenotype. And what it gives? Ovaries, abdomen, and not abdomen alone. It gives attitude also. So your attitude you can make positive or negative depending on your epigenome. See, if you stay with a positive individual who has a positive thinking, your thinking becomes like that after a few months. But you are staying with a person who is highly depressed mode, you will also become like of a depressed individual. Why so? Because this is just you are changing your own epigenome. So your attitude, if you want to qualify or do anything, you have to stay in a highly positive attitude and interact more with those individuals than with an individual who always try to bring negative thoughts in your mind. So queens contains more chromosomes, interactions, and more. This is not to go this we talk about out of chipset. And people have shown these are the two modifications which happen on them and they regulate their gene expressions. So now, till now what we have understood till about environment. So how these environmental factors affect us? That is very important to understand. So growth factor, hormones, cytokines, different nutrients. They undergo different metabolism, signal transduction, transcription factors, and they all modify this chromatin. And all the tags which I have just now talked to you. What are the tags we talked about? Acetylation, methylation, phosphorylation. So you know where from this acetyl comes? Any thoughts? No thoughts. I will show you a thought. So when you eat glucose, one of the substrate which is produced is called acetyl-CoA from the citrate. So this acetyl-CoA acts as a substrate for histone acetyl transferase enzyme and brings about the acetylation. So how this is nicely linked with the metabolism, if you are doing, we say yoga, we say exercises, help us. How they help us? More of oxygen is going into it. You have a glucose, your cycle is becoming more towards glycolysis and TCA, and you are producing more of acetyl-CoA in your own body. And that acetyl-CoA is responsible for opening your chromatin, and that is the natural habit of an individual to have a more open chromatin organization. Then there are other substrates like NAD, NADH. Then there comes alpha-ketoglutarate. And there's another pathway which is very, very important, which is called S-adenosylmethionine, SAM, which is responsible for any kind of methylation in our body. So what is that pathway? That is called one-carbon metabolism. That is called one-carbon metabolism. I will not go into any detail of it. It is basically folate, vitamin B12, and the vitamin B6 pathway, which brings about at this state production of S-adenosylmethionine, and which is a substrate for DNA methylation to happen. And the best known example for this, if I quote you, any pregnant mother when goes to a doctor, the first prescription is folate and vitamin B12. Why so? You must have heard about it in your own family around. Anybody who is pregnant goes to a doctor, the doctor immediately like start folate and vitamin B12. Because they need a more production of these things to happen. SAM. And why SAM is essential? That helps in condensing a chromatin. And when the chromatin is condensed, the cell division is slow. And therefore, the cell gets more time for repair to happen. And this is one of the aspects of it. Another aspect which is very important in developmental biology, in the uterus, the conditions are highly hypoxic. This is only 2.1% oxygen when the embryo is developing. And that again is required for a slow division of cells to happen so that the errors are not accumulated during the course of developmental phenomena. Because we know at every DNA replication there will be errors, and these errors will be corrected in the G2 phase of cell cycle. And therefore we maintain highly hypoxic state, and we always prescribe folate and vitamin B12. And these are very well shown in the neural crest defects. Also in the congenital heart defects, how the levels of folate and vitamin B12 correlate nicely with the early embryonic development. And why we are seeing, because the pregnancy generally by 1 month you get to know, by time your neural crest and the cardiac development has finished. And if you have a low level of folate, there's a tendency to develop these diseases. So these studies have been shown and they are very nicely emerging also in the field. How these metabolic pathways or the metabolites play a critical role in changing your epigenome, and therefore different developmental defects comes out there. And so therefore, if you see, if you have a right level of this, there's effective methylation. If you have a deficiency of choline, betaine, folate, or vitamin B12, there will be a defective methylation happens. And this associates with, today, with multiple diseases, today what we know. So now if I come down to all the things, another concept has emerged which is called exposomes. What you are exposed to, I was talking in between here and there. If you see, it's an ecosystem, lifestyle we talked about, but the most important is social. Household income, we compare. The families compare between the two. Inequality, social capital, social network, cultural norms, cultural capital, psychological and mental stress. We always forget these phenomena. These are very well we talk about. Contamination and others. But comparing with two, I have a Maruti 800, somebody has a Mercedes. I want to purchase a Mercedes car. So that brings a complete stress in a family, and that stress completely changes your own behavior. So these earlier in a society, if you see 100 years before, we are not talking about depression. 50 years before. Why we are talking too much of depression today? Among the research scholars or the student communities, somebody is having a mobile of 30,000, a lakh. I have a mobile of 5,000 rupees. So I will be start living in a stress phenomena. My mobile megapixel is 20, others is 40 megapixel camera. Why? How it matters in life? Why you have to compare with anybody? Enjoy your own life, what you have, and have a fun out of it. And this, if you don't do it, you will suffer with different kinds of epigenetic alterations. But the problem with epigenetics is it tries to sustain you for a few years, but if your habit doesn't change, then it leads towards mutation and further diseases. I will give you a nice example on this. So this is a nice example I will give you here. When we move towards a little bit of cancer initiation, there are genetic alterations, but we know about there are BRCA or PSA, there are different mutations which causes different kinds of cancers, but everybody having those mutations don't suffer with the disease. We talk about BRCA, we talk about colorectal association of APC, we talk about hepatitis B virus infections, but everybody don't lead towards this disease until unless there is a priming happening in between. And this epigenetic priming leads towards a more further secondary mutations, and that it leads towards a developing cancer. Similarly, it happens in the susceptibility when we talk of SNPs. The priming and environmental factors are key to cause the driver mutations. It is not that if you have a mutation, you will lead towards a disease. So if you see in a colorectal APC mutation, if you are having a very good dietary fibrous diet, then you don't suffer with a colorectal cancer. The studies existed. So therefore, you are taking a lot of pressure on the colon for absorbing a water. So this is all the factors which are involved. And if your whole genome is perfectly okay, there's no SNPs, but the environmental factors, persistent exposure to a bad environmental causes a change in the priming. Cell try to resist itself, try to adapt, but that is always a limit. Once that is not happening, then it causes a mutation leading towards a cancer development. So till here, everything is under your control, whether you want to revert it to this phenomenon. And the best known example when we talk about, I will give always about a gastric cancer. We talk of atrophy, hyperplasia. We all suffer with, if we have a heavy food, we will take Pandy or Gelusil or something, and we have the gastritis and other kind of a problem. But if you take a drug, you become normal in few days. But generally irreversible or chronic irritant persists, then it leads, these phenomena leads towards. All these phenomena are highly reversible. So it is us, if we are taking those kind of bad food on the roads, then we suffered with these kind of diseases. And that's what the example I gave you of the roasted and a chicken and other things in the beginning. So those bad foods, irritants. So these are the irritants to our GI tract. And when the irritants are there continuously, they lead towards the disease. So with all these fundamentals, now understanding epigenetics, in 2022, there's a paper published in Cancer Discovery where 4 hallmarks have been added. Non-mutational epigenetic reprogramming has become a most important hallmark because it dictates unlocking phenotypic plasticity, mutational epigenetic programmed innate senescence cells, and microbiome alters your own epigenome. Therefore, people say always, if you see, go back to little 30, 40 years, the curd was a usual dietary material in our own meals because that gives a good kind of a bacteria for our own stomach. And that was very, very important to understand. Today, we don't eat all those kind of food. We don't eat curd and other things in a regular meal. And these were giving the good microbiome to our own things. And this microbiome regulates a lot of phenotypic alterations in our body via epigenetic mechanisms. So now a little bit of, I think I've talked about fundamentals. I talked about a little bit on this. Already this is 12. So we give a glimpse of what we do. So I will show you the gastric cancer. If you say 5 to 10% of a gene are associated with development of gastric cancer, 90 to 95% are environmental factors. And what are those? Infection of persistent expression, bacterial infection of Helicobacter pylori, smoking and alcohol factors, salt intake, family history, vitamin C intake, nitrosamine and nitrites. I've talked to all of you. And what happens when these patients suffer with this, have a bad habit and they have a gastritis or a gastrointestinal problem? What the usual factors? Gelusil, Digene, or any other kind of a drug, which is given on a counter sale. You suffer with the disease until unless it becomes an advanced or at least T2 or 3 is passing, then you come. And when you come at that stage, the problem suffers are high locoregional recurrence, poor response to surgery, poor response to chemo, delay in presentation, and there's a high relapse of cancer happens. These are some of the very standard phenomena, and therefore, there's a very, very poor prognosis for these patients. These are some of the work we have published. I will not go into that, but I can just show you, to do a science, what I'm talking to you in just hardly 10 minutes, almost 1, 2, 3, 4, 5, 6 students have worked for almost 5 to 6 years to give me what I am going to show you. See, when the surgery is performed at Tata Memorial Center, the tumors are removed. We know exactly from the site of tumor what is the resection margin. These distances are also measured at Tata Memorial Center and we know how it is happening. So whenever I am talking about resection margin, the RM is mentioned. We profile different histone modification in these gastric human cancer tissues. We profiled in almost around 110 human cancers way back in 2014. Shafqat did all this work. And then what we came out, the acetylation is decreasing. Methylation overall globally is not changing. But what we saw, the phosphorylation is significantly increased. I will not be talking about, so this was a very nice observation, very interesting to us. But any clinical significance we can build up. Phosphorylation, we built up a very nice significance. And we showed it can define the true cut margin for the patient. And at least in GI at Tata, what we defined them, if the distance from the tumor is around 4 to 5 centimeter, the loco recurrence is almost negligible. And that the data, and so if they get 4 to 5 centimeter, the first scissor runs from there for removing the tumor tissues. But the interest was, we saw acetylation. What is happening? What I talked about acetylation in the beginning, if you have a higher level of acetylation, the chromatin will be in open organization. But what is happening? The acetylations are lost. And if the acetylations are lost, chromatin will get highly condensed. That is what is happening. So that's what we observed. And so this was a very nice Western blot and ISC, we confirmed it. We confirmed at a levels of ISC, also at RNA level. But these are the change in acetylation may be happening because of the two enzymes. Either there is a decrease in histone acetyltransferase or increase in histone deacetylase enzyme. So these are two enzymes. And enzymes increase in the level of protein doesn't make any sense until and unless the activity of an enzyme is playing an important role. Because level may be more, if the activity is not there, it will not do any function within a cell. So any enzymes when we talk about, it's always the function of an enzyme which plays a role. So we, and what we found is a significant increase in the HDAC activity. And the HAT activity almost remains the same, suggesting this pathway below 1 is getting more active here. So this way, the normal open chromatin, which should be always the normal cells have a normal open chromatin, it is getting condensed in a gastric cancer. So what sense we can make out of it? So this is what the scenario, a nice balance on a seesaw, but because of an HDAC, we have a highly condensed chromatin in a tumor cells. That's what we came out very nicely. So what we thought of, there are a lot of chemotherapeutic drugs like epirubicin, cisplatin, oxaliplatin, which are routinely used in clinics for treatment of these gastric cancer patients or different cancer patients. So what we thought of, if the chromatin is highly condensed and these drugs bind to DNA, the binding, because the DNA is condensed, the binding will be very, very less. What we thought of, if we can treat these patients with an like this, and if I can open and throw the drug, more drug will bind and the cell death will be more. So that was the basic concept we came out. There are a lot of clinical trials have been done, have been reported to failed in solid tumors. If you go to clinicalgov.in, when we were analyzing our own samples, we found out on 110 samples, RNA is almost higher in 74%. But when we came down to the activity, it is only 24%. And if I treat all the 100 patients with HDAC inhibitor, it will show positive, may show a positive result in 24, but the toxicity of 76 will fail my statistics analysis. And then I will say my trial has failed. And that's what is there in the trials have been done on the basis of that. So we started looking in this concept. We started treating with an HDAC inhibitor in a cell culture, and we could find really the cell kill. If you see, these are different ways of treatment. When I give an HDAC inhibitor before, the cell kill in a culture was significantly high with any drug or any of the HDAC inhibitors. We went ahead, did an animal study. We find when we did VPA followed with cisplatin, there's a tumor. It's simply much, much smaller in comparison to other treatments, suggesting there is action happening. This was the concept we came, but the problem happens there always. When we leave these tumor animals which have been treated with VPA and cisplatin, the tumors are very, very small. But when we leave them, the tumor comes back again. After 10, 15 days, if you see, and if you see all the research papers today, what they do, they treat, regress, publish. We left these animals for around 2 weeks, and 70% animals shows the tumor is coming back. So what the use of relapse? So this was a relapse happening after the therapy. So we thought of what is happening there. And this phenomena, I think, who are working in a field of microbiology must know drug persistence, antibiotic drug persistence cells. But when you treat with any of these bacteria with the drugs, some of the cells survive and these are called drug persistence cells. Same concept emerged somewhere around to 2020 in case of cancer biology. So what we did, and these cells are thought to be only transcriptionally reprogrammed. People have shown by single cell data sequencing, there are no mutations acquired in those, suggesting epigenetics may be a key player into it. So we made the drug persister cells here, drug tolerant persisters, we make it in culture. And we did some of the analysis, which I will not go in detail. So when we were looking into what we found out, the tumor cells have a condensed chromatin, what I showed you earlier, but the drug-tolerant persister cells has a much, much higher condensed chromatin. And then resistant cells again have a open chromatin organizations. And this we saw the changes in the heart activity has decreased, HDAC have gone up in the drug-tolerant persister cells. So similarly, we did for the methylation. We saw it's highly condensed. If it is condensed, methylation should be high, and that's what we all observed. So we thought of give different HDAC inhibitors and different histone methyl transferase inhibitor independently and see what happens. We saw a similar phenomena again, but if you leave again, they come back again. So that was quite frustrating to us. So this paper we published recently, a synergistic action of HDAC inhibitor with cisplatin impedes survival and proliferation of drug-tolerant persister cells in gastric and liver cancer cells. But then we wanted to know if this is happening, what any other phenomena is giving advantage for these drug-tolerant persister cells to survive. So you all must be knowing what is the major organ in the body which provides you energy. Mitochondria. So then we thought if the mitochondria has to do something with it, we went because mitochondria is the organ which is providing what I talked to you just now, a lot of substrate for our epigenetics to happen. So we went ahead and did just I would like to highlight here, this is a parental tumor cell. The mitochondria are smaller. In a drug-tolerant persister cells, we have a big mitochondria. Resistance again has a small mitochondria. Clearly suggesting there's a mitochondrial biogenesis happening. We go deeper, we showed number increased, copy increased, all those things we did. We told the mitochondria fusion is happening, and that is forming. So all this data came us to one conclusion: the OXPHOS pathway is becoming more active in the drug-tolerant persister cells. Whether that holds true in the field of macrobiology, not known in literature. So that is a very interesting problem which anybody can quickly do in the labs and look into OXPHOS pathway. So this came into our picture. And then we looked into ROS antioxidant balances. So what we showed drug tolerant, it has a high OXPHOS. So what we can do now? So we thought of why not to target OXPHOS pathway directly. And there's a very good drug which you can repurpose. It's a known diabetic drug, metformin. And metformin is used in clinics and there is no toxicity observed. Another drug for epigenetics, so we use metformin. Another drug for the dual epigenetics, we used valproic acid, which also routinely used in a clinic for epilepsy. It is given for 330 years to different patients, and they have not shown any side effects. So these two drugs, when we used, and together in combination with cisplatin, and you will appreciate the last line where I gave VPA and metformin together, the treatment stopped here. The arrow shows the stoppage of treatment. And then there is a tumor is not coming back again. We have followed these animals for a much longer period, almost around 2 to 2.5 months, and they have not given us back tumor, suggesting it is not a single drug which is required, but you need a multiple drug to act on tumor. And the doses what we are giving here are the of metformin, IC20, what is given to the routine diabetic patient. We are giving IC20 of this, and cisplatin is IC50. So we have decreased the dose as a synergistically they are acting together. And that's what we came out, minimal residual. So if in a cancer, the problem is always of a minimal residual disease, the disease which remains after the treatment. And we could little bit work out on that, and we have found a very important results. So chromatin modifiers and metabolic pathways inhibitors sensitize cancer cells and make them vulnerable to therapies like chemotherapy and radiotherapy. So therefore, now what we have is a phenomena, what we all talk about, personalized medicine. We can't treat every patient with metformin and valproic acid. I have to stratify the patients on the basis of who have a higher HDAC activity, have a higher OXPHOS. And that was a purpose. Maybe I don't have a slide on that, but we have developed a protocol where we can measure the levels of HDAC and HAT activity in a serum samples. And Therefore, also we can measure the levels of OXPHOS enzymes. And now from the serum samples, I can subcategorize a patient on the basis of high VPA, high HDAC activity, and high OXPHOS activities, pathways. And those patients, what we want to take ahead, and that what we have now taken ahead, madam, with all the support in the beginning, to another CSR funded project. This project was literally funded by a CSR, which is very difficult to get out, coming from a coming from a bank of Japan, MUFG funding, of a good funding for there, where we are taking this study now for bringing the proof of concept, give all the patients who have a higher HDAC activity assays, which are non-responsive. We have done the studies almost around in 30 patients till now. And out of that, around 12 patients have high HDAC activity and they're non-responsive to the treatment. This is in gastric. In rectal cancer, we have another story going on where a Dr. Reena, an engineer from TM, she has given this project to an MD student, which was quite supportive to us. Okay, a clinician is getting interested. She has just taken all the patients from the rectal cancers and we did an IHC on that. All class 1 HDAC. All the patients, what she came out, were non-responsive to the radiotherapy, have a higher levels of HDAC3. And that was quite surprising to us. So now from a 50 sample study of an MD student, we have started with a 1,000 sample study and trying to bigger a cohort and see whether we can take it to the clinics. So basically what I brought down today to all of you, the gene function when you talk about epigenetics is incident. Nutrition, stress, and environment are the players. Where we have to go ahead into the field where the future directions lies, epigenetic and metabolic variation are an important consideration. For personalized, we talk about different levels of mutations for personalized treatment, but these need to be assessed now. Dynamic epigenetic biomarker for health status, we don't know today. We have very few. So these are the problems which need to be taken by the youngsters who are sitting here and try to define for us. Clinical application, understand the connection between exposomes, what I showed you, and the genome behavior, how this happens there. And their center part in our own body is metabolism. If we can define the metabolism, which is our totally unworked field, people are never interested to read the pathways. They don't like reading pathways in biochemistry, but every science lies here in the metabolic pathways. And people have to study much into deeper to make the sense out of it. So therefore, what I say always, this we know very well. This we are trying to understand. We are also. But if you see how genome interacts with the environment, that gives us epigenomics. This has nothing to do. Epigenomics gives us transcriptomics. Transcriptome gives us proteome. And the functional proteome gives us a metabolome. The sequence has to be made to go deeper into field and to differ everything. Thank you all for patient hearing. And thank you for giving the opportunity. Perfectly okay.